All right, so are you ready to dive deep into receptors?
Let's do it.
Awesome. Those tiny communicators running the show inside our bodies, they're what make your heart race when you see someone you like.
Or tell your muscles how to move when you're, you know,
Dancing.
dancing, right?
Yeah, or even, like, help you taste that perfect slice of pizza.
Pretty much the ultimate control panel, right, for our biology.
Exactly. But you know what? These receptors,
They can be pretty weird.
oh, they can be seriously strange, leading to, like, some wild phenomena.
Yeah, like we're talking mutations that give people basically superpowers.
And even, like, those accidental drug discoveries.
Yeah.
The ones that completely revolutionize science.
Oh, and don't forget those times when evolution just went nuts with cellular communications. Like, what were you thinking, evolution?
Seriously. You want to be the life of the party.
This is the deep dive for you.
Absolutely.
Yeah.
So let's start with those superpowers. You know, maybe not flying or invisibility, but some receptor mutations are pretty darn close. Ever heard of the EP4 receptor?
The EP4 receptor. Sure, that's the one found on cardiomyocytes, the cells that make up heart muscle.
Right. So think about it. You have this receptor hanging out on a cardiomyocyte, just waiting for its special delivery.
And then boom, prostaglandin E2 shows up.
Prostaglandin E2.
Yeah, it triggers this whole chain reaction.
Like a domino effect?
Exactly. It's called the G's cascade, which basically ends up with the cell-making cyclic AMP.
Okay, so far so good.
Right.
But here's the wild part. This same G's cascade, the exact same chain reaction, also happens when a different receptor is activated, the beta-1 adrenergic receptor.
Oh yeah, the one activated by adrenaline.
You know it. The stuff that kicks in when you're about to, like, give a big presentation or run from a bear.
Right. So both the EP4 and the beta-1 adrenergic receptor, they both activate the same pathway, the G's cascade, but
Totally different outcomes.
totally, like stimulating the beta-1 adrenergic receptor, that increases heart muscle contraction, that classic fight or flight response.
Okay, but what about stimulating EP4?
Not much happens. It doesn't have that same effect on heart muscle contraction.
Wait a minute. So two different receptors trigger the same cascade, but one makes your heart pound,
Yeah.
and the other one's just like, whatever.
Pretty much. That's actually what led to the discovery of something called cyclic AMP signaling microdomains.
Okay, microdomains. Is that like, what, tiny VIP sections inside a cell?
It is. It's a great way to think about it. Imagine the cell is a city, and these microdomains are like different neighborhoods, each with its own vibe.
So even though both these receptors, they activate the production of cyclic AMP, the effects of that cyclic AMP depend on which neighborhood it ends up in.
Precisely. Like, think about having the same ingredient, cyclic AMP,
Okay.
but using it to make, I don't know, a spicy curry in one neighborhood and a delicate souffle in another. Same ingredient, different outcome because of the environment it's in.
Oh, that's a great way to put it. So it all connects back to those superpowers, right? Different receptors, they trigger the same cascade.
Yeah.
But the effects depend on which microdomain, which neighborhood it's happening in.
That's a fantastic way to think about it. These microdomains let cells fine-tune their responses even when they're using the same signaling pathways. It's pretty incredible.
So basically, it's like having the same key,
Yeah.
but it can open all these different doors in a giant mansion.
Right.
And behind each door is like a totally different experience.
Exactly. And this whole concept of microdomains, it's completely changed how we think about cellular communication.
It's mind-blowing. All right, so we've got these superpowers, right? All thanks to microdomains. But let's shift gears and talk about those accidental drug discoveries, sometimes those breakthroughs.
Oh, they can happen in the most unexpected ways.
Seriously. So this story starts with some scientists studying the effects of certain drugs. Just normal science stuff. But then they stumble upon something totally weird. These drugs, they were messing with the activity of a key player in the cyclic AMP signaling pathway, this enzyme called protein kinase A.
Or PKA for sure.
PKA, got it.
And it turned out these drugs weren't actually targeting PKA directly. They were interacting with these other proteins that were like clinging on to PKA. And now we call those proteins A-kinase anchoring proteins.
AKAPs. What is it, like a funky 80s band?
Haha, maybe. But in the world of cells, they're more like stage managers for PKA, making sure it's in the right spot at the right time to do its job.
Okay, so AKPs, they're like the scaffolding
Yes.
that holds up a building during construction.
Exactly. They act as scaffolding inside the cell, anchoring PKA enzymes in very specific locations. And just like with those microdomains, location is everything.
Okay, so I'm picturing these AKPs, and they're like super organized, keeping everything tidy and in its place.
Exactly.
But how do they bind to PKA? Is it like a secret handshake or something?
Not magic, but there is a special mechanism. Think of it like a lock and key. AKPs have two main parts. One part is the targeting domain, and that's like a GPS telling the AKP where to go inside the cell.
Like a cellular GPS.
Okay, and the other part is the PKA binding domain, which obviously grabs onto PKA.
Okay, so one part's a GPS, and the other's like a little hand reaching out to hold on to PKA. I love it.
That's it. And within that PKA binding domain, there's this special structure called an amphipathic helix. Imagine a magnet. One side sticks to metal, the other doesn't. That's kind of like the amphipathic helix. One side loves water.
And the other side's like, no way.
Exactly. And that picky magnet, the amphipathic helix, it binds to very specific spots on the PKA regulatory subunits, anchoring them in place.
So it's picky about where it hangs out.
Absolutely. Oh, and did I mention there are actually different types of PKA, like type 1 and type 2?
Yeah.
And they have different preferences for AKPs.
Hold on, different types of PKA. So it's like having different types of construction workers. Some want to be way up high on the scaffolding, and others are happy closer to the ground.
Perfect analogy. Type 2 PKA, it loves hanging out with AKPs, while type 1 is more independent.
Interesting.
And this difference, it affects how they function inside the cell.
Wow, it's like a perfectly choreographed dance, all happening inside our cells. We've got these superpowers in microdomains, and we've got these incredible AKPs acting like, what did you call them? Stage managers.
Yeah, stage managers.
Making sure everything's in the right place at the right time. It's amazing. But are you ready for things to get really weird?
Oh, I'm always ready for weird.
Let's dive into evolutions, like greatest hits of bizarre adaptations when it comes to receptors. What have you got?
Well, have you ever heard of the glycine receptor?
Glycine receptor. Doesn't ring a bell.
Well, it's kind of a big deal when it comes to, keeping our movements smooth and controlled.
Oh, okay. So we don't end up like looking like robots gone haywire.
Exactly. Our muscles are always getting these signals to contract, but we need something to stop them from going overboard. And that's where our friend, the glycine receptor, comes in.
Okay. I'm intrigued. Tell me more.
All right, picture this. You've got these alpha motor neurons in your spinal cord. They're the ones responsible for, making your muscles contract.
So like the gas pedals for our muscles.
Perfect analogy. But you don't want to have the gas pedal stuck down all the time, right?
Definitely not. That sounds like a recipe for disaster.
Right. And that's where the glycine receptor swoops in to save the day. It's like the brakes.
The brakes. How so?
Well, it sits on these alpha motor neurons. And when a molecule called, you guessed it, glycine comes along and binds to it, it actually stops the alpha motor neurons from firing.
So it's like a safety mechanism.
Yeah.
Like to prevent us from, I don't know, accidentally crushing something with our superhuman grip.
Precisely. And it's actually pretty fascinating when you compare it to what goes on in the brain.
Oh. What happens in the brain?
Well, in the brain, there's this neurotransmitter called GABA. You might have heard of it.
GABA. Oh, yeah. Isn't that the one that, like, chills everything out?
Exactly. It's like the brain's chill pill.
So are you saying glycine is like the spinal cord's version of GABA?
In a way, yeah. They're both inhibitory neurotransmitters, meaning they help calm things down. Glycine's the traffic cop in the spinal cord, keeping our movement smooth, while GABA keeps the peace in the brain, helping us relax.
Okay, that makes sense. Different security guards for different parts of the city. But how does this glycine receptor actually work, like on a molecular level?
Well, it's what we call a ligand-gated ion channel.
Ligand-gated ion channel. That's a mouthful.
It's basically a fancy way of saying it's a gate. It opens and closes in response to a specific molecule. In this case, that molecule is glycine.
Oh, okay, so glycine is the key that unlocks the gate. What happens when the gate opens?
Well, when glycine binds, the gate swings open, and these chloride ions, they flow into the neuron.
Chloride ions. Okay, and what do those do?
Think of them as tiny tranquilizers for the neuron. They make it less likely to fire, sort of like turning down the volume on a loudspeaker.
So to recap, glycine binds, gate opens, chloride ions flow in, neuron chills out, muscles don't go crazy.
Right. You got it. It's all about maintaining that delicate balance between excitation and inhibition. It's crucial for our muscles to function properly.
That's pretty amazing. Evolution really came up with a clever system to fine-tune our movements. But I remember you said things were going to get weird. What other evolutionary marvels do you have in store for us?
Well, how about electric fish?
Wait, electric fish? As in fish that can, like, shock you?
You bet. Some of them, like electric eels, can generate these incredibly powerful electrical discharges. We're talking up to 600 volts in some cases.
600 volts. That's enough to power a small appliance. How's that even possible?
They have these specialized cells called electrocytes that basically work together to create these electrical pulses.
It's like they have a built-in battery pack.
It really is.
Yeah.
But the really mind-blowing part is how they can sense those electrical fields.
Yeah, do they have like some kind of built-in voltmeter?
Even cooler than that, sharks and rays, for example, they have these organs called ampullae of Lorenzini.
Ampullae of, what was that again?
Lorenzini. Ampullae of Lorenzini. Basically, these tiny pores filled with this jelly-like stuff that can detect even the faintest electrical signals given off by other animals.
So they're like living, breathing metal detectors, but for electricity.
Exactly. They use them to find prey. Even if it's hidden under the sand, it's incredible.
That's seriously impressive. But how do receptors fit into all of this electrifying and electrosensing?
Well, scientists believe these electric fish have evolved these modified ion channels. They're super sensitive to any changes in electrical fields.
So it's like evolution took those gatekeepers we were talking about earlier, the ones that open and close to let ions in and out, and gave them this like electric upgrade.
You got it. These special ion channels, they're not triggered by molecules binding to them. They're triggered by changes in voltage. When they sense an electrical field, they either open or close, letting ions flow in or out, and that change in ion flow. It triggers a whole cascade of events, which allows the fish to either sense or generate electricity.
Wow, evolution never ceases to amaze me. But I have to ask, why should we care about electric fish? I mean, they're cool and all, but how does this relate to us?
Well, it's a perfect example of how evolution can take something basic, like ion channels, and modify it to create something totally new and incredible. And by understanding how these systems work in other organisms, we can gain insights into our own biology and even get inspiration for new technologies.
So it's like studying nature's blueprint for innovation.
Exactly. Nature's been experimenting and innovating for billions of years. There's a wealth of knowledge waiting to be discovered.
This whole conversation about evolution has got me thinking about like how receptors came to be in the first place. They seem so fundamental to life as we know it. Do we have any idea how they first evolved?
Well, for that, we need to take a trip back in time, way back, billions of years ago, to the very first cells on Earth. Back then, life was a lot simpler. We're talking single-celled organisms floating around in the primordial soup, just trying to survive.
Wow, we're going way back to the very beginning. But how did these early cells even communicate?
It was all about sensing and responding to changes in their environment, things like temperature, pH, and the availability of nutrients. They probably relied on simple mechanisms like changes in their membrane permeability.
Membrane permeability, so like how easily stuff can pass in and out of the cell.
Yeah, precisely. Imagine a door that can open or close depending on what's going on outside. If there was a sudden change in temperature, the cell's membrane might become more permeable to certain ions, and that would trigger a response to help the cell adapt.
Like a basic alarm system, letting the cell know something's up and it needs to take action.
Exactly. But as organisms became more complex, they needed more sophisticated ways to communicate and coordinate their activities. So those simple alarm systems evolved into specialized proteins that could bind to specific molecules, like hormones, triggering much more specific responses.
So those ancient alarm systems basically became the complex receptors we see today.
Exactly. Evolution over billions of years has shaped and refined these cellular communication systems, leading to the incredible diversity of receptors we see today.
It's mind-blowing to think that something as complex as our nervous system, with all its billions of neurons and trillions of connections, started from those basic communication mechanisms in the primordial soup.
It really shows the power of evolution to take simple building blocks and, over vast amounts of time, create something incredibly complex and beautiful.
I don't know about you, but I'm feeling a sense of awe right now. It's like realizing we're all part of this grand evolutionary story, and these tiny receptors are a crucial link connecting us back to the very origins of life.
Yeah, it really is amazing to think about how these tiny receptors, they've shaped evolution, and they're still playing such a crucial role in our lives today.
And the most exciting part is that we're only just starting to understand them, their full potential.
Oh, really?
Yeah, scientists are actually looking into new ways to kind of manipulate receptors to treat diseases, even enhance human abilities. Okay, now you're talking. So we could potentially create like drugs that target specific receptors to say block pain signals.
Exactly.
Or maybe even like boost the immune system, kind of like giving our bodies a software upgrade.
That's the whole idea behind targeted therapies, deliver treatment right to the source of the problem.
And fewer side effects, hopefully.
Exactly. And there's also a lot of talk about using gene editing, to actually repair or modify those faulty receptors.
Gene editing, huh? That sounds straight out of science
I
fiction.
know, right? But it's advancing so quickly. And it holds so much promise for treating diseases that we basically thought were incurable.
It's incredible to think about, but it also makes you wonder about the ethical side of things.
Oh, absolutely.
I mean, if we can manipulate receptors to treat diseases, could we also use that same power to, like, enhance human abilities? Like, create super soldiers or athletes with enhanced senses?
That's the big question, isn't it? Like with any powerful technology, it's crucial to have those open discussions about, the potential benefits, but also the risks.
We don't want to accidentally create a real-life
Oh,
Frankenstein.
exactly.
But it is exciting to think about how understanding receptors can help us, I don't know, unlock new mysteries about ourselves and the world.
Absolutely. For example, there's some evidence that receptors might actually play a role in how we perceive the world, like influence how we experience beauty or even spirituality.
Really? So like those feelings of awe and wonder that we get when we look at a sunset or listen to a piece of music, those could be influenced by receptors.
It's a fascinating area of research. Some scientists think certain receptors in the brain might be involved in, like, mediating those transcendent experiences.
Wow, that really makes you think. So it's like those tiny receptors aren't just controlling our bodies, they're shaping our consciousness too.
It really highlights how deeply intertwined biology is with our experiences, how we perceive the world.
Well, I think my brain is officially full of receptor knowledge. This deep dive has been amazing. We've covered so much. Superpowers, electric fish, the origins of cellular communication. Who knew something as small as a receptor could be so fascinating and impactful?
It is remarkable, and I think the biggest takeaway is that we've only just scratched the surface. There's still so much to learn about these incredible molecular machines.
So to all our listeners out there, if you're looking for something to really dive into, I highly recommend exploring the world of receptors. You'll never look at the world the same way again.
And you never know, you might even be the one to make the next big discovery.
Exactly. Keep those questions coming. Keep exploring. Stay curious. Thanks for joining us on this mind-blowing adventure into the world of receptors.
Until next time.